So with this, I think I will be asking Dr. Andres to go on with his presentation. You're welcome, sir.
Okay, thank you so much. I just wanted to apologize for all these postponements and also thank for inviting me to give here this presentation. The idea in this presentation I developed here is just to give an insight quickly my path to bioinformatics, because I initially studied the molecular biology, and then on my path in my career there was a switch going over to bioinformatics, and now I'm a bioinformatics specialist. And then I want to look into what the work we do at IITA. So on one side, the bioinformatics we do, but to show you how this is connected to the general research at IITA and what is the role of bioinformatics in the general research in an institute like IITA. So I have to see. So here you just see some lines, places where I've been. So I'm Swiss, and as I said, I was studying at molecular biology at the Swiss Institute of Technology, and at the same institute I did my PhD, always as a molecular biologist. Then I had the opportunity to do my postdoctoral research and experience in University of California in Berkeley, where I continue working as a molecular biologist in plant research. Then I moved back to Switzerland and had first a postdoctoral position at a Novartis Foundation. So this is a research center which was is sponsored by Novartis, the pharmaceutical company Novartis, where I continue to do molecular biology, but then I got involved in new technologies like microarray. This was the first high throughput technology, where we produced a lot of data, and we realized that we need to start to do what we call bioinformatics. So we have to write software to analyze this data. So this was the time when I was transitioning from molecular biology to bioinformatics. And the transition was kind of smooth. So it happened that I was working during the day in the lab, and in the evening, late evening, until sometimes even almost the whole night, I was writing programs to analyze this data. And at a certain point, I realized that this is not really life. So I had to decide whether I stay in molecular biology or I go to bioinformatics. And there was then the decision to go for bioinformatics. And I developed from then on my skills as a bioinformatician. Then I got a position in the Italian Research Council, an institute for biomedical technologies. So we are doing mainly development on new tools and databases to analyze data which come from human research. And in this background, I started to specialize myself on the analysis of high throughput sequencing data. At this Institute for Molecular Biomedical Technologies, I have a permanent position, but I had the opportunity to do a sabbatical in Nigeria. And there I was offered a position, kind of a researcher position, to develop a bioinformatic environment. So I was 3 years in Nigeria, in Ibadan, developing this bioinformatic infrastructure, and then later you will see what this means. And right now I'm mainly back again in Italy, and I continue as a consultant to maintain this bioinformatic environment and train and help to analyze biological data at IITA, and of course other institutes which are collaborating with IITA. When I talk about IITA, of course, it's not only Nigeria. IITA is also in other countries all over Africa, and I'm involved in all regions in Africa to train and educate in bioinformatics. So the team right now shrunk dramatically, but we are still a competitive team with 2 collaborators in Nairobi. This is Trusha Shah and Michael Landi. And at Ibadan is myself and Temitayo, which works mainly as system administrator and helps in bioinformatics. I trained about 7 MS students, and right now Temitayo finished end of last year his PhD and is looking for his postdoctoral position to continue in bioinformatics. The collaborations are all over the world, but the main interaction we have, of course, is the University of Ibadan, because it's close by, and since ever IITA was actually founded 50 years ago, was interacting with this University of Ibadan. But we have also close interaction with the Bioscience of Eastern and Central Africa, very close interaction with the Swedish University of Agriculture in Sweden. Then from my times in Switzerland, I still have very close contacts with the University of Fribourg in Switzerland. And then as a big partner in the States is Cornell University and Boyce Thompson Institute. So with help of these collaborators, we develop new solutions and analyze the data. Bioinformatics side, having biological data, of course, means that we need informatics, and informatics means that we have a computing system to do all our calculation. So we right now are lucky owner of a server which has 1.5 terabyte RAM, so we can really deal with huge data amounts coming from the sequencing, these next generation sequencing technologies, and then of course attached storage that we can maintain this data. You can imagine that if you start with a big data set, this means that during the time of analyzing the data, you produce more and more data. So in the normal case, you quadruplicate your data during the analysis. So you can imagine that storage, and especially fast storage, is a very important part of your analysis. We have 3 other servers which are less potential, but we still have quite size of RAM to serve for smaller projects, and especially also for teaching. So having a group of specialists in bioinformatics and having this infrastructure on the hardware side, we are quite a potential force in bioinformatics at IITA. And just to mention, we are more than welcome to interact also with other institutes. So we are collaborating, we can also help. And what we right now are doing more and more is, of course, training. So if there is a need in training in bioinformatics, I'm more than open to discuss and see whether there is a possibility to help in this issue. Now, I just would like to focus on some points, and I just want to pick out these things which I'm mainly interested in. So my major interest is regulation of gene expression. To look at that, we generate 3 different data types. So the first, of course, is the expression levels of the genes. We need to know this. And then we look at data which we hope to understand how these genes are regulated. And these are the small non-coding RNAs and the DNA methylation. And I will go into this a little bit more in detail during explaining the projects I'm working in. So one of the projects which I started from the very beginning, I joined IITA, is to understand what mechanism we have, what mechanism Cassava developed to be resistant against Cassava Mosaic virus or the Cassava Mosaic disease. This topic is still in investigation in also very famous universities in the US and Europe, but up to now, there are no clear answers what makes a plant resistant. And this would be very important to understand for breeding and also for helping to convert farmer preferred varieties which are susceptible or not highly resistant to get them resistant, that we don't lose the properties that the farmers prefer or the end consumer prefer, whether this is how it cooks, how it is the texture, taste, all these parameters. So we did a SNP analysis of about 5,000 genotypes, and we were able to localize a Q12 on a chromosome. It's 12 here is written scaffold, but meanwhile we know that this belongs to chromosome 12, and we were investigating in the beginning this region of this QTL to understand whether we can find out what is in this region which makes the difference between susceptible and resistant. So we were sequencing leaves of plants which were diseased and susceptible, and plants which are resistant. In some cases, we have some viruses. In some cases, we don't have virus. And we were analyzing this data to extract genes which might be responsible for this difference. Small RNAs are non-coding RNAs which has a size of about 21 to 24 nucleotides. Here you see the blue line is a small RNA, which is generated by transcribing from the genome, and then it gets processed in a molecule, which here you see as a red line, which is the small RNA. And this can interact with the transcript with a messenger RNA, either to initiate the degradation, and this is a way to regulate the expression of a gene, because if we lose the transcript, we cannot produce the protein. Or it can also attach to certain regions of a messenger RNA and block the production of the protein. So small RNAs are important regulators of whether a transcript or a gene transcribed in RNA will be at the end produced into a protein. The other data I mentioned is a DNA methylation. DNA methylation is actually a way in life developed also to regulate the expression of a gene. You have on positions on the genome at the cysteine, you can have a modification where a methyl group is attached. And this methyl group doesn't change actually the sequence of the DNA, but changes from a sterical point of view, the attachment of a polymerase to the DNA. So if you have these RNAs, this DNA methylated, there is a probability that you cannot, the system cannot read anymore the gene and cannot produce RNA and later protein. In this graph, you see different sequences, what we did. So you see again, we have susceptible cassava, which is either virus-free or is infected. And we have a resistant cassava, which is virus-free or infected. And if you look at this graph here, you see these blue bars show how much DNA methylation you have at this region. And we observe, and this you see from what I described here, you see that the susceptible, which has no virus, has at this region a region which has very little methylation. Whereas the resistant, whether this is virus-free or is infected, and the susceptible, which is infected, has here increase of methylation in this region. So this means the virus somehow triggers in these plants that they methylate this region. So from this information, we can imagine that the virus, the plant is reacting on the virus with this methylation to try to protect against the virus, and it gets a similar state than the resistant plant. But most probably, and unfortunately, we didn't find yet the site. There are some of these sites on the DNA, on the genome, which we don't have this shift from low methylated to high methylated, like at this position. And these are most probably the regions which are making the difference whether the plant is resistant or susceptible. And then you can imagine we integrate this data. So we compare the small RNAs we analyzed and how they interact with the transcripts, the messenger RNA. We also see how smaller RNAs are interacting with this methylation profile. And of course, we will understand how this methylation profile is influencing the RNA production in this plant. So you see, we have 3 different data sets which will be integrated and should give us an answer of why some cassava plants are resistant and some plants are susceptible. So this data here, we hope we will get information on genes which are involved, and this will help the breeders to find the markers for better breeding of farmer preferred cassava genotypes. And on the other side, we will have some mechanistic information to see whether we can work also with transgenic plants to change this problem of resistance. Another project is Striga in maize. I don't know whether you know, Striga is a beautiful flower which is growing on the roots of the maize plants. The maize on a certain point dies or will not produce enough seeds to with a drop of yield in these maize fields. We again set up an experiment where we have a resistant genotype and a susceptible genotype, and then both of them, we infested them and we did not. So we have here the control of the plants which are not infested with the seeds from Striga, and we have the plants which are infested. We checked 3 different time points: an early time point, an intermediate, and a very late time point where Striga starts to grow and come to the surface. Again, we sequenced the RNA of these samples, and what we can see here by this graph, we can see that we are nicely able to separate the transcripts from the resistant plants to the transcripts of the susceptible plants. So it is clear that some regulations in the susceptible plants are clearly different than the regulation in the resistant plants. So searching in this data and comparing these infested and non-infested samples and the susceptible with the resistant samples, we were able to find genes which are clearly regulated in favor of the resistant plant. And one is genes which are involved in the lignin biosynthesis, which means a cell wall. So it seems that the resistant plants have a way, a mechanical way to resist against the attack of these Striga plants to enter into the root and start to benefit from the metabolic activity of maize. On the other side, we found a pathway which involves a component which is actually an antifungal compound. They found out in leaves of many plants that this is produced in leaves against fungus, against bacteria, against feeding from insects, but never was observed that this compound is also produced in the root. And we managed to demonstrate that this compound, if we add this to our culture, inhibits the growth of the seeds of the Striga. So we managed to individuate this compound, which is called Timboa, and we found out that it is very simple to demonstrate how much of this Timboa we have in the roots by a colorimetric analysis. So you see the tubes which have a dark blue have a lot of Timboa, and tubes which have less strong color, they have low Timboa. So like this, we can analyze in a breeding project, we can analyze very early and understand whether the roots are producing this Timboa or they are not producing this Timboa. So with this project, we were able to give to the breeders an additional tool to very early select the offspring from a crossing, whether these plants are resistant for Striga or not. And another project, again on cassava, we wanted to understand the development of the root system. And what we can see here in these pictures is that you have some roots which are dark and thick and some roots which are light and thin. And we classified them as potential storage roots, which are the dark and thick one. And the other one are fibrous roots. Fibrous roots are important for water and mineral uptake. And of course, the potential storage roots, they are responsible in a certain time point to store the starch and grow to the roots, what we at the end will eat. So we wanted to understand whether we can differentiate the activity and the gene expression between these two root types. So we again were setting up expression analysis, having 4 tissues. We took as controls leaf tissues from early young leaves and older leaves. And we took, of course, the samples from storage roots and fibrous roots. We have 3 time points. The time point is a very early time point, a time point shortly before the storage roots start to bulk, and a time point clearly after bulking to get the most important actions in the development of cassava. And then, of course, to do statistics, we need biological replicates. Again, we were able to differentiate the expression profile between storage roots and fibrous roots. And we were able to demonstrate that storage routes from the early beginning have a totally different metabolomic pathway in starch production. So all these red arrows show that these genes which are involved in the storage production and the starch production and the starch storage are upregulated already in the second time point in the potential storage routes compared to fibrous routes. So we were able to demonstrate that there are already genes in these routes which are differentially regulated according whether this is a storage or a fibrous route. The other thing what we managed to do is to differentiate, I apologize, to differentiate on one side comparing the leaf profiles against the root profiles, root specific genes, so genes which are only expressed in roots. And then when we compare storage against fibrous roots, we can find genes which are clearly differentially regulated, whether this is a fibrous root or a storage root. So again, we managed to demonstrate that these two organs are clearly differentially developed with different genes involved. In collaboration with externals, this is the Swiss Institute of Technology, we were analyzing the sugars, the soluble sugars and starch. And of course, here you see the orange line is the sugar and starch content in storage routes, and in blue in fibrous routes. So clear different metabolic profile in these two routes. And even the starch granules which we produce, we can show that here up in the fibrous routes, there are different starch granules than when we look at the different time points in the storage route. And I just added this slide here just to demonstrate what possibilities we have, and bioinformatics actually goes further in image analysis. Here we have experiment with Fraunhofer Institute in Germany, where we manage with X-ray to follow the growth of cassava in a pot. So you see this is an image we took, a three-dimensional image we took at 24th of August. And 2 months later, we took the same plant, and we can see that some of these roots, like this one here, this became a storage root, while this root here was staying most probably a fibrous root. And this technology, here again, the same thing, we can see over time how the roots are growing without destroying the plant. And what we were able, if you reconstruct three-dimensionally this information from the X-ray analysis, you can make virtual sections through these roots. And we were able to demonstrate again here that this root here, which later we demonstrate became a storage root, has here an inner channel which is connected to the vascular system of the stem. And even very early, not visible by eye, but if we do a profile of the gray levels, we see that there is a drop of gray level here, like we have here. And again, we can demonstrate that these roots are very early becoming storage roots. And this is, again, very important for breeding in cassava to understand that we can monitor very early how many storage roots a plant may produce and influence then the yield of this cassava. Actually, this was published in case somebody's interested in plant cell and environment, the whole work we did on this root morphology. And then last but not least, a small research project, which is not really related directly to any of the topics at IITA, but just a technological analysis, is that technology of RNA sequencing is more and more used, but is still quite an expensive technology. And what you see here, I demonstrate this, that is the gene in red. And then you produce from these two exons, you produce the RNA. And when you do a traditional sequencing of RNA, you will sequence the whole stretch of RNA, and then you have to reassemble the whole thing and produce the information you want to have. There is a new technology which is called 3' RNA sequencing, where in this case we only fish the last fragments of these fragmented RNA sequences, and we sequence, in this case, 1 sequence per transcript. So if I have 4 transcripts here, I will have 4 reads which are coming from the sequencing, whereas in this case here, we maybe have 20 reads. So this means I produce less data, but I also have to pay less because the sequencing machine has to produce less reads. And if you map this information to the genome and compare these 2 sequencing technologies, and it's more or less this thing represents what I showed here. Here you see in blue is the structure of a gene. The fat blue lines are the exons, and the thin ones are the introns. So you see, if I do a traditional RNA sequencing, I will have some sequences coming from the fat parts here and here and here. In some cases, there's nothing, or here again. Whereas if I sequence 3' RNA, I have only 1 peak at the very end here, which represents the last sequences of my RNA sequence. And you see just here visually, these are much less reads I need to understand how this gene here is expressed. And this gives me a dramatic a decrease of cost. And right now is about 5 to 6 times cheaper, which means I can 5 to 6 times sequence more different samples. We are now developing an algorithm to analyze this data. And just to point out problems we have, so we have in our RNA-seq peaks, which are coming in the middle of the RNA. So we have to evaluate whether this is an artifact or whether this is maybe an alternative end of this gene. We have peaks which are outside of what is annotated. And in case of Cassava, the annotation is still in progress. So it could be that this is not perfectly annotated, and this gene is going all the way down to this peak, which we don't know yet, and we are analyzing this. Here, just some statistics about first analysis we did. So if you see the 3' UTR means the very last piece of our RNA sequence, we see that on 15,000 of these annotated regions, we have no sequence. We cannot find sequence. We have genes, almost the same amount of genes which have not an annotation for a UTR. And we see that we hit with this RNA-seq only 10,000 of the 40,000 genes we find reads which are coming from this region. And the reason why this number is so low, you see in here, if we move out of our annotation downstream our genome, we see that some of these RNA-seq are actually mapping way down. So meaning here, these peaks here are peaks which we find, meaning that probably many of these annotations are not yet correct. And with help of this RNA sequencing, we can re-annotate the gene structure of Cassava. So this, I hope, was not too much overload on you. Is the peaks of our research, the Cassava, the bioinformatics group in IITA. I'm open for questions. Here is also my email address. If you have later questions, or you have bioinformatics related question, or maybe a request for some help, you can anytime contact me, and I will try to get back to you as soon as possible. Thank you.
Okay, thank you so much, Dr. Andres, for a nice presentation and what you have shown to us today. I will be asking those who participated, if you have any question, to send it in the chat while I start with my own question. Then in absence of any other further question, then we could also close. Now, I observed your interest in the gene expression amongst all of these plants with several challenges. So I just wanted to ask in general, what is the end point of this research? For example, in the cassava mosaic disease, where you are making some observation of what could be responsible for the resistant and also for the susceptible. So which problem do you clearly want to solve at the end of the day if this your research progresses very fine?
Yes. So as I demonstrated with the cassava, especially with the mosaic disease and with the Striga maize, the aims can go in two directions. One is producing information of genes which are involved in certain traits. In my case now is the resistance against cassava mosaic virus. If I found these genes, I can produce markers, and then the breeder can have a marker-assisted breeding to accelerate the breeding process. So I have very specific information with very specific markers, and the breeder can see immediately in the very early stage whether this gene is expressed, which he needs that the plant is resistant for the virus. On the other side, in Striga, I demonstrated on one side again, if I know that I need Timboa in my plants, that the plant is resistant against Striga, I can produce markers, and actually we are working on this, and I hope soon they will test them. But on the other side, you saw I was even able to propose a way how they can much faster find out whether it's Timboa there. They don't have to isolate DNA and then sequence for SNPs and then see whether the SNPs of these markers I propose are there or not. They just can squeeze a little bit of root, make the reaction, the colorimetric reaction, and then they see if it's blue, Timboa is there, and means the genes are expressed, and means that the probability that this plant is resistant against Striga is very high. So we have mainly our results go to the breeders and help them to be more specific in their way to select what is for the traits they're interested. And we can help them even to find easier ways to find this out. So they don't have to go through a SNP analysis, which they do right now, because they have SNP markers, which they relate to traits. I can offer, in this case of Striga, a much easier approach. And theoretically, this approach is they're even able to do at the same seeds, which then they will put into the field. So they grow the seeds, remove one of the roots, make the assay, and if this plant has a blue color, then they put this in the field. If the plant doesn't have a blue color, they trash it.
Thank you. Now we have one more question here in the chat from Aminu Abudlai. How is it possible that plant pathogen to induce mutation? I don't know if you understand that. Not very clear to me.
I see. I have to say, I didn't say that the virus are producing mutation. Virus can, I guess this refers to my DNA methylation. So this is very important to understand that the DNA methylation is not a mutation. The DNA methylation is a reversible modification of the environment of the DNA. So my sequence stays the same, but at the cysteine there is a methylated methyl group added, which gives you a sterical, so a different form. When the polymerase arrives to the DNA, it cannot recognize anymore the DNA, and is not able to transcribe this region. So it's not a mutation, but it's an alteration of the DNA. And the virus is able to do this because, and I didn't go into this, but it's quite a complex mechanism. The virus itself is able to trigger a system to produce small RNAs. And I talked about these small non-coding RNAs, because the plant uses these small RNAs to fight the virus, but the virus is able to use the same small RNAs to try to get influence on the plant. And the influence on the plant is that these small RNAs are going on the DNA and changing whether it's methylated or not. It's very complex, and a lot of these steps are still not known. But we are working on this, and actually we have quite a big project sponsored by the Swedish government to analyze these things.
Yeah, thank you again, Dr. Andre. Another question from my own end, which seems to be, so in what platform do you generate your sequences? Currently, we know about the nanopore sequencing, so I wouldn't know if this has found application in the IITA environment.
Right now, we are not using nanopore sequencing for two reasons. One reason is that the analysis we are doing on let's say orphan crops like yam, maize is not anymore orphan crop. What did I say? Cassava is an orphan crop on the level of the bioinformatics. I'm working, for example, also in yam. So we need precise sequence, and nanopore is not yet giving the precise enough sequences that we can analyze them and understand about the sequence. What we are using is mainly Illumina, because this is the most reliable, but unfortunately very short. But for RNA analysis, this is good enough. And we use PacBio for some resequencing of genomes. Right now, we have also a project where we produce some reference genomes for cassava, African cassava varieties, because the official reference is a South American cassava, and we see that there are quite some differences between these two genomes.
Okay, thank you. And maybe a follow-up question is, I mean, the Illumina and the PacBio you mentioned, do you work with the other collaborators outside Nigeria, or you have these platforms in the...
No, we don't have the platform. No. Maintaining such a platform is very, very time consuming. No, it's very cost consuming. And we decided to not have the sequencing environment in IITA, but we use providers in Africa, but also in the States, in Europe, whatever. I mean, we always scan through and see who gives us the best offer. Because the prices are dropping, and it is worthwhile to do over and over check how much they want to charge. I mean, RNA sequencing was from $500 per sample, dropped now to $250. And probably continue dropping. So it's always worthwhile to ask different providers, because the problem is not a big problem. You just isolate your RNA and you send this to them, and then they sequence and send you the sequence back. The effort to maintain a sequencing platform is huge. I mean, if you don't do this very professional, I mean high throughput, then it's not worthwhile. If you can guarantee that you will have a lot of customers, then you can try. But I didn't want to go into this. You have good service groups all over the world which are doing the work for you.
I see. Yes. So I think, yeah, we have come to the end of today's webinar.
Yes.
We're overshot by a minute or so. And once again, I want to thank you, Dr. Andres Giselle, for coming and for at least making sure that today worked for this presentation.
Yes.
And we have learned a lot also, and some other applications that probably there will be some personal follow-up at the background through your email that you have sent.
I'm open to to help out. Just take in consideration that I will not reply in 10 minutes, but I will reply. I'm more than happy, and I'm again, thank you. I mean, it was my pleasure to present for you guys. Yes.
Thank you.
Okay.
Thank you, and take
care and have a nice
Yeah,
afternoon.
and thanks
Bye-bye.
for the audience that participated. We thank you for
Yes.
coming, and they will follow up and send the recording of this meeting across to your email with which you registered for this webinar. Thanks and remain blessed. Bye.
Okay.
Yeah.
Bye-bye. Take
Yeah.
care.
Yeah.
Thanks.
Yeah.